Started. All right, so today I'm going to talk about a little bit of work that I've been doing on the side on protein structure prediction using AlphaFold, which you may have heard of, and I'll introduce. And those are running using Parcel, and it's also in collaboration on a DOE grant that I've been working on with Parallel Works Inc. And so before I get started, I just want to thank my group, a lot of whom are using Parcel for their various projects. And I talked about two of their projects the last 2 years, including Daniela, who's an undergrad who graduated from NYU recently, who's done some of this work with Parallel Works. And I also want to thank people at Parallel Works, including Mike and Stefan and Matthew, who helped on this workflow, and Shenlong at our HPC, who set up AlphaFold for us. So to just introduce the problem briefly, there's what people call the protein folding problem, which is that you have a string of amino acids which somehow have to fold into some shape, and that shape gives it its function. And in general, it's a hard question to ask when or why do you have a certain set of amino acids fold into a certain shape, but you can run simulations of that. There's a separate problem called the structure prediction problem. And a lot of people say, when they say they've solved structure prediction, they're saying they solved protein folding, but it's not necessarily true. The question here is to take a one-dimensional string that describes this polymer and predict what it would fold into without any care for how it turns into that shape. Just a bit of history, which is nice full circle thing, is that I was working on this problem as an undergrad at UChicago when I was working with the Swift team, and we even have an unpublished manuscript about doing this with Ian and Mike and so forth. So that's one reason I'm still very interested in this problem. So you've probably seen some of these news stories like the greatest achievement in AI ever for AlphaFold. But what is it if you haven't been following it? So there's, like what I was talking about before, there's a semi-annual competition to blind predict the structure of proteins using whatever method you can come up with. And this year, the team from Google DeepMind, led by John Jumper, who's a UChicago graduate, produced this new deep learning model called AlphaFold 2, which predicted almost everything with almost 100% accuracy, which was amazing. Just to mention that David Baker's group at Washington, after they heard the method of AlphaFold described in the conference, they produced their own version without seeing the AlphaFold code. It's called RosettaFold. So this is another deep learning model with extremely high accuracy. But what really got everyone excited was that, unlike 2 years ago when they had a good model, AlphaFold and also RosettaFold published their code on GitHub, and you can just use it. So that means that anyone can do this. So what does that mean? I would say before 2021, what you had to do was, in my field, I should say, I'm not interested in the protein folding problem specifically. I'm interested in what do the proteins do. And so if you want to do a simulation model of a protein, what you had to do was first check if it was in a database of known structures, and then you could do an MD simulation, a molecular dynamics simulation of it, and study it. If not, you had to pick a different project. So that was kind of lame. So now I would say what you can at least try is you can take your protein, run it through this deep learning model, and then you can run an MD simulation, at least give it a shot. So what are the challenges of this? That AlphaFold is pretty computationally expensive, not super computationally expensive, but it requires a lot of data to do the full model, like 2 terabytes of stuff. And it has some scripts that I don't think are all that elegant, but you can't just say like, oh, I want to predict all these structures and run simulations of all of them. So that's what we're trying to set up here. And so what we're trying to make is a workflow that looks like this. You have a bunch of files for your different proteins. Currently, we're using the Parallel Works workflow manager to do this. So we then use that to distribute a Parcel job to either NYU HPC or Google Cloud. The AlphaFold has been implemented by Shenlong, our HPC person, in a Singularity container, which I've talked to some people about. So it's actually implemented exactly the same way on Green and HPC. On Green and GCE. However, I've been using a version where you don't need the full 2 terabytes of data because that causes some problems. Anyway, so from this, we can make many predictions with different random seeds for each one, and then we can feed that into something to set up MD simulations and then fire those back off onto more HPC jobs. So for those who haven't seen the ParallelWorks platform, which you'll see later, what you can do is build a form so that someone who's a novice can basically upload the files for each protein and just hit execute, and it will run on either Google Cloud or HPC or other resources. The parcel script for this is really trivial. It says loop over all files and run AlphaFold on that file. So perhaps overly simplistic compared to maybe what you'll hear about later. And one of the nice things about the ParallelWorks platform is they built this result explorer so that you can actually browse your results, which I may show in a minute, live for a second. Oh, I guess I can do that now for a quick second. I think I have about a minute or so. So, so the results of, for example, running this thing look like this, and you can sort of sort them by some quality metric, and they're also colored by that. And you can click on them, and we also just integrated an ability to browse the result in 3 dimensions. So with this thing, you can kind of explore the different results of your outputs very easily, which I think is an advantage of, I don't know, being organized in advance or something like that. So finally, I'll just conclude in what I learned from this project. So first of all, use of singularity, and then just having a different bash script for each type of resource made it really easy to just switch from cloud to supercomputer. But I wanted to kind of pose the question of whether app definitions per site still make sense, like we used to have in Swift, in a sense of like not running different. I know you can decorate it in parcel tasks, but kind of having like a different script that is actually executed by a bash executor, for example, on different sites that you tag it with that. I don't know if that's a feature that's useful for everyone, but throw that out there. Using the ParallelWorks platform was nice because we get this visualization and also plug in your cloud credentials and things. So that was nice. But the main reason that I think it's nice is this click button thing. So I could imagine using this in my class next semester where everyone picks a protein, uploads a thing, pushes a button, and then has a simulation. But I should note that people have built Google Colab notebooks where you can do this one protein at a time all the way through starting an MD simulation. So that's also cool. And finally, I noticed that this very simple workflow might be a good case for FuncX, which I think I had already gotten an email from Ian and Kyle about that. But I did think that what's interesting is as you go on to do more visualization analysis and starting other simulations, that maybe parcel starts to make sense again. You can tell me if that's not the case. Okay, that's it.
Great. We've got a couple of minutes for questions. I guess stick a message in the chat if you have a question. I'll start. You said that you just at the end you were talking about platform-specific app definitions like Swift used to have. Is that something you've experimented with in Parcel, or is it just a pie in the sky? Wouldn't this be nice idea?
So what I've done is this is something that may be interesting to talk about, but what I have done is have my Bash app take as one of the inputs a script which executes the task. And so that script is staged to the remote machine. And then depending which site I want to use, I have a different script that knows, what path to execute in or how to load Singularity or whatever. So that's the way I've currently been doing it, which doesn't require anything fancy, but it makes your parcel script longer, right? So if you kind of knew in the same way that FuncX is set up, if you kind of knew like running GROMACS, which is something I want to do, that GROMACS executes through Singularity on this machine and it uses different Singularity with AMD GPUs on this machine, if that was kind of set up in a database or something that you could make, then that would be, or a file database like a YAML file or something, then that would be potentially useful, I think.
Okay, cool. And in the chat, Matthew has a question. Do you want to unmute and ask it?
I can answer this. So yeah,
yeah. So the question is,
I didn't know I had to vocalize my question.
No, yeah. So the question is whether AlphaFold separates folded and unfolded domains. So it tends to apparently do a really, so it has the structures I showed are actually colored by some kind of machine learning metric that they've figured out, which says like how confident they are in each residue. And so it tends to be very confident in folded areas and unconfident in the other areas. So it's working surprisingly well. And something I didn't have a chance to mention is people have been hacking around with this thing. And AlphaFold can do all sorts of things that they didn't plan on, which like if you give it the sequence of one protein and then a random linker region and then another protein, it will actually predict how two proteins bind to each other because it's, I don't know, because it's learned so much information about how residues are close to, or protein amino acids are close to each other. So that's really good. The only weakness it seems to have is a couple of things in membrane proteins. And I don't know, it's remarkably good. And I think we can all expect that, in 2 years, it'll be even better.
Yeah, well, the membrane proteins, one side of the membrane has a completely different chemical condition than the other. But yes, cool. Thank you. Yeah.
Yeah, and they actually get some membrane proteins correct because they are known structures of that too. So
cool. Okay.
Good.
So on to our next